Hey, everybody. Welcome to yet another Journal Club session brought to you by Lifespan.io and Life Extension Advocacy Foundation. I'm Dr. Oliver Medvedik, and today we have something special. We have Alexander Tyshkovskiy joining us as the lead author of the paper that we're going to be covering, or he's going to be doing most of the covering. It's a pretty heavy-duty statistical analysis paper that well, it's in Cell Metabolism. I'll allow Alexander to introduce himself shortly. The paper we'll be discussing is identification and application of gene expression signatures associated with lifespan extension. So just like the title suggests, in this paper, they looked at a large number of data sets, primarily from mice, looking at RNA sequencing analyses, as well as other data sets, and looking at essentially expression and other profiles that correlate with lifespan extension through a variety of different interventions, pharmacological, dietary, and genetic backgrounds of mice and genetic interventions. So basically trying to figure out what do all of these interventions that extend lifespan have in common? And then developing a tool that researchers can use. I think it's called Gentervention, pretty nifty name, that researchers could apply to their own data sets to see if there's a high probability of a particular intervention extending lifespan. And I think this is really cool and a really useful tool for researchers because, we do genetic screens, people do screens for lifespan. I mean, you have all these interventions. There's so many things you can do. And we're always looking for biomarkers of aging. So in a sense, this is sort of like a really robust, I don't know if I should call it a meta biomarker, but this is aggregating a lot of data and coming up with a profile that would suggest, strongly that a particular intervention, whatever it is, pharmacological, dietary, I mean, whatever it is, if you look at the expression profile and it matches, the profile that's using the Gentervention tool, will it have a high probability of extending the lifespan of an organism? So having a tool like that is, to screen through all of the myriad interventions you can do, both singly and in combination, is very, very useful. So what Alexander here is going to go over is basically the paper that laid the foundations for this, this tool that researchers could use. And there's a number of important insights that have arisen from all of the data analysis that's been done here. So a little bit, in case you were wondering, I'm not in the Cambar Center right now. I'm in the kitchen of my apartment, and we are broadcasting actually pretty close. This is actually my girlfriend's apartment, and we'll be moving out shortly. But in the meantime, we're using this as the temporary headquarters, and we're actually pretty close to Cambar Center. So geographically, I'm a 15-minute walk from work, so pretty close to Cambar. But anyway, without much further ado, I'd like to, welcome Alexander Tyshkovskiy.
Yep.
Dr. Alexander Tyshkovskiy.
Almost a year already.
Oh, wow. Okay.
It was my thesis project, by the way.
Nice. This is a very awesome thesis project, by the way. So congratulations. This is pretty amazing, and a lot of great authors on this paper as well. So anyway, welcome to the Journal Club.
Thank you, Oliver. Hello, everybody. Just a small remark before we begin about gene intervention. So gene intervention allows you to look at the association of each individual gene with lifespan extension. Currently, it doesn't allow you to run the whole association analysis to predict how, the whole intervention of interest associates with longevity. So we are right now developing this new application to do exactly that. Gene intervention for now just allows you to look at single genes and visualize their associations. For example, you think that certain gene can be linked somehow to lifespan, and you can easily go there and just look at the gene of interest, immediately get picture for publication. That's the idea. Okay, so I think now we can proceed with the paper, right?
Sure. Wherever you want to begin, or if maybe give a a brief introduction, or and take it from there.
Yeah. Okay. I probably can share my screen.
Mm-hmm.
Yeah. So what is it about in general? Well, Oliver already told the idea. So the general idea was to find biomarkers of longevity, basically biomarkers of lifespan extension. Because now we already have pretty big portion of data related to systemic effect of different interventions on different biomarkers such as gene expression, which is probably the most popular systemic high throughput data type. And so the idea was to look which genes are consistently associated with lifespan extension across different interventions, regardless of the type of interventions. You may have a diet, you may have a certain mutation, for example, growth hormone receptor knockout. You may have different drugs such as rapamycin, acarbose, etc. So they may have different targets, molecular targets, but the idea was to look at the whole state of the tissue, how they change the whole state, and which changes are most consistently associated with lifespan extension. So this was the idea of the project in general. And as a model, we took mice because, well, they are the closest relatives of human across typical model organisms. So we started our research from our own data. First picture gives some overview. So we had 8 different interventions, and in green here on panel A, you can see interventions that are new. It means that there has been no data on them previously. And in the most cases, we had both males and females, and they also look at different ages. Well, basically the main reason was just the data we had. I mean, we couldn't really choose, but the big representation of ages gives us also a possibility to look at most consistent changes, meaning that, I mean, regardless of the age when intervention started, or it still may have some robust biomarkers. So we had the data, and immediately from this data, we already could see some similarities. So we could see them at the level of individual genes shown on panel B. Here we demonstrate caloric restriction in males and females, snail dwarf mice. This is a mutant of growth hormone synthesis. And methionine restriction, which is another interventions related to longevity. And you can already see from there that lots of genes overlap, differentially expressed genes, I mean. So at the level of individual genes, we have some common mechanisms. The same can be seen at the level of functional enrichment. If we perform gene set enrichment analysis, then we obtain panel D, and we see that, for example, genes corresponding to ribosome proteins are upregulated across almost all interventions. And here, each bar demonstrates individual interventions and also individual sex. So they are upregulated across both males and females across different interventions. Another common biomarker is the upregulation of cytochrome P450 and glutathione metabolism genes, and also oxidative phosphorylation. Others are more specific. For example, you can see fatty acid oxidation may be upregulated or, on the contrary, downregulated, depending on the type of intervention. So this is more specific mechanism. So here we thought that it's already a good start. So we may aggregate more data, because here we have, as I said, 8 interventions. We want more. So, and we already have available data in database, public databases have been published by our groups. So we wanted to aggregate all the data on mice together. And that was our next step. But yeah, I think before that, just small interesting part, which is not the main idea of our paper, but we found it pretty interesting. This is related to feminizing effect. So it is well known that some interventions, such as caloric restriction and growth hormone receptor knockouts, they somehow feminize gene expression and concentrations of certain proteins in males. So it means that the concentrations of these proteins become more female-like after introduction of interventions. And we wanted to see if this pattern is universal, if it's specific only for certain interventions or it's general pattern. And for that, we first identified differences between males and females, which is pretty simple. And then we looked how intervention change these sex-specific genes. If they change them in the same direction, in the opposite direction, or, I mean, in no direction with correlation of 0. And panel B shows this effect at gene expression level. So here we observe that indeed GHRKO, which is growth receptor knockout, and snail dwarf mice, which is another growth hormone related mutant, both of them significantly feminize male gene expression profile. So the expression of sex-specific genes in males becomes more female-like. And the same is true for diets, not only for caloric restriction, but also for methionine restriction. And interestingly, feminizing effect occurred not only in males, but also in females. So they became more female-like, even though they are already females. However, for our interventions, there was no significant feminizing effect. And for some of them, there was even significant masculinizing effect. So the expression became more male-like. For example, we observe it in many cases in females, for example, in response to rapamycin or 17-alpha estradiol. So it seems that based on this data, feminizing effect is neither sufficient nor required for lifespan extension. And the same was basically reproduced using metabolome data. So we used metabolome as validation of this observation, and we basically demonstrated the same thing. However, interestingly, what you can see from this panel, from both gene expression and metabolome data, is that in all cases, except for just one case, which is protandim, in all other cases, males had more feminizing effect than females. So it means that the expression of genes which are more activated in males, in controls, they became more female-like in males compared to females. So basically, it's kind of hard to imagine, but what is, I mean, easiest interpretation is that in response to certain intervention, regardless of the type of intervention, males and females gene expression profiles are kind of moving to the same common state. So these sex-specific differences, they diminish in response to lifespan extending interventions.
So two questions, Alexander. So one question is looking at the data there. You may have this information or not because it's not what you looked at here. But some of the interventions, like you see, like for CR, for example, there's clearly a feminizing effect, or some of the genetic backgrounds, like the growth hormone receptor knockout mice, clearly a strong feminizing effect.
Yep.
Some not so much, like rapamycin A-carbose, which lowers blood glucose levels. is there a correlation? Off the top of my head, I don't know, but looking at, I don't know if they've done it with these mice. These are, I think, these het3 mice, correct? That's the data,
Yep.
these heterogeneous mice. Is there a correlation between the degree of feminization and lifespan extension that you get with these interventions? So the more feminization, the better the intervention works for lifespan, or is there no, or has anybody not even looked at that?
So in general, where there will be a correlation just because growth hormone mutants are usually the most long-lived mice. I mean, it's the most the strongest intervention that you can imagine
Right,
now, and diets have also pretty strong effect.
right.
But if you look specifically, then it turns out that it's not the how to say, it's not the real biomarker, because, for example, in case of proton-dim, it extends lifespan only in males,
Mm-hmm.
but in males you can see lower feminizing effect than in females. And well, for example, for rapamycin, you have the opposite picture. It works mainly in females, but here in males, it's more feminizing. So there is no consistent dependence, I would say.
Right. And on average, you said that you sort of get like looking at the data in figure B. I mean, I guess on average you get I guess I don't want to say regression to the mean, but you get a basically females get more masculinized, males get more feminized, I guess. But that's only for certain interventions, right? I mean, some of them it's clearly you have feminization happening even with female mice. So when you said that you get the statement that you had a consistent let's see. I guess just to clarify that, is that just like an average that you get that you get, I guess, the sex effects normalized, or is it just for,
Okay.
or is it depends on the intervention?
Yeah, talking about moving to the common state, I mean,
Yeah, yeah,
diminishment of
yeah.
specific sex-specific differences, it's shown on panel C. So
Mm-hmm.
basically the difference between males and females. Uh,
Uh-huh.
every point here is pairwise distance between two samples,
Okay.
one male and one female sample. So all pairwise combinations. And in red, you have control mice, and all other points basically show different interventions. So you can see that in all cases, except for proton-dim,
Mm-hmm.
there is significant reduction of distance between males and females. Only in case of rapamycin applied for 12 months, we see non-significant decrease. It's a bit smaller, but not significant. In all other cases, it's decreased, and there is a significant decrease. And the same we can see at the level of metabolomes. So it seems that it's kind of consistent pattern.
Mm-hmm.
Of course, there are some exceptions, of course. So some of circles are higher than others, but statistically, we see such effect for every individual intervention except for proton-dim for some reason.
Mm-hmm.
So when we looked at the particular genes involved in this feminizing effect, we basically obtained panels F and G. So here you can see which genes are involved in this feminizing effect. It means that these genes are, first of all, differentially expressed between males and females, and second, they are perturbed by feminizing interventions, right? So they are correlated with feminizing effect. And if we look at pathways enriched by these genes, we observe drug metabolism on the upside, and on the downside, complement coagulation cascades and major urinary proteins. And what is interesting here is that almost all of these pathways are known to be regulated by growth hormone. So it seems that the majority of these genes involved in the feminizing effect can be explained by a single molecule, basically. So we believe that this can be an explanation of this pattern, and it is not really associated. I mean, functionally, it may not be associated with lifespan extension, but it's correlated with this effect usually just because lifespan extension also involves uh, change of growth hormone levels. So it's kind of confounding variable here. It's both involved in lifespan extension and in feminizing effect. So yeah, this was part related to feminizing effect. Now I will return to biomarkers of lifespan extension. So as I said, we aggregated publicly available data, and panel D shows the data we had in the end. Now there are 17 different interventions, and they were obtained across 77 different data sets. By data sets, I mean comparison of control versus certain intervention. Some data sets were from the same sources, from the same articles, because, for example, in our case, we analyzed 8 different interventions just in one batch. But in other cases, the number were different. But in total, there were 77 different comparisons. And we had all types of interventions. We have diets, we had drugs, we had genetic manipulations. So we had pretty good representation of different types of interventions. First thing we did, we looked at biomarkers related to specific interventions, for example, to caloric restriction, to rapamycin, and to growth hormone deficiency. The main reason for that was basically just to validate the method, because caloric restriction was previously analyzed by some groups at meta level, I mean by aggregating different datasets, and we wanted to see if our results basically were well correlated with the results of our groups. And also based on that data, we also saw that some of the functions are again pretty well shared by different types of interventions. And here basically all the data are independent. So it's nice that all of them are shared. So for example, oxidative phosphorylations once again is upregulated across different types of interventions. So this is kind of a consistent effect. But when we aggregated our interventions, so in total we had 17 different interventions. Oops, sorry. Yep. And first thing we did, we just looked at the similarity between different interventions. How similar in general is their effect. We're talking now not about individual genes, but just about global gene expression effect. To do this, well, we had different approaches, but the main unbiased approach that we did was to look at pairwise correlation coefficients between different data sets from different sources. That's the main point. So if you just take 2 interventions from the same source, then you basically have shared controls between them. And this is not independent comparison. That's why if you just do comparison between datasets from the same source, you will have on average higher correlation than you have just independently. So this is biased approach. To make it unbiased, we looked only at comparison between different sources. And so here panel B shows the results, the result of this analysis for caloric restriction. So the idea again was simple. We took all caloric restriction datasets we could find, and we ran all pairwise correlation tests with our certain our intervention. And basically, these box plots, these violent plots, show the distribution of correlation coefficient. First of all, this is a nice thing that caloric restriction correlated with itself. So this is kind of validation. Caloric restriction is number 5 here. So on average, the coefficient was about 0.4. So again, this is the correlation coefficient for comparison of data sets from different sources. This can be different sexes, different ages of mice, different dose of interventions. So all possible designs you can imagine. So on average, the correlation coefficient is 0.4 if you take the whole gene expression profile. This is for caloric restriction. And the same you can do for our interventions. And basically, we can sort interventions based on how close, how similar they are to caloric restriction. And then the idea is to do it for the whole, for all comparisons of interventions you can imagine. And this is panel C. So the same analysis is done for all versus all. And here every square represents median correlation coefficient between every pair of interventions. In some cases, we have gray boxes. It means that we had no available data sets from different sources. So, for example, resveratrol and every other day feeding, which is EOD, they have data sets from the same source. That's why we couldn't compare them with each other in an unbiased way. So that's why there are gray boxes here. This is the correlation matrix, and what we immediately can see here is that, first of all, there is very tight positive cluster of growth hormone deficient related interventions. So we have Ames dwarf mice, little mice, non-dwarf mice, and growth hormone receptor knockout mice. All of them basically lack growth hormone or growth hormone receptors. So basically in the liver, and all of this analysis is done in liver. In liver, this converges to the same, basically to the same effect. So liver just doesn't sense growth hormone. That's it, and that's why the correlation is very high here. After them, we have diets. They also have positive correlation with each other and with growth hormone mutants. So they are also kind of positive. But for other interventions, you may have distinct patterns, and the most interesting case was the case of rapamycin, which is believed to be caloric restriction mimetic, but in our analysis it showed significant positive correlation only with itself. So in all other cases it was distinct based on the effect on the whole gene expression profile. So that was pretty unexpected. Well, in general, we see that on average, interventions have similar effect on gene expression. So on average, the correlation is positive. And that's why we were interested in identifying common signatures of different interventions. So basically the biomarkers of lifespan extension, which genes responds to the same, in the same direction, regardless of the type of intervention. And we basically identified such genes. We identified about 300 of them, and it's shown here. So we did it again, both at the level of individual genes and at the level of functional enrichment. So at the level of individual genes, it's shown on panel B, for example. This is CTH, which is cystathionine gamma lyase, which is involved in the metabolism of methionine and in production of H2S. And it was basically very well known to be involved in the effect of methionine restriction, not surprisingly. But here we found that it's consistently upregulated in response to very different interventions. So it's shown here on panel B. It's shown first aggregated within each intervention. So for example, we take all data sets corresponding to caloric restriction, and we kind of aggregate them together, look if the gene is upregulated robustly across different data sets. And we did it independently for each intervention, and basically this figure shows for each interventions how these genes react. On the lower panel, we have the same shown for each individual data set. So here every color represents certain intervention. So for example, all yellow dots are caloric restriction data sets, and basically the upper panel shows how these individual points aggregate together in like aggregated response. After that, we did functional enrichment analysis, and we observed some of the functions that were already familiar to us, for example, drug metabolism and oxidative phosphorylations. These two functions were top two. So basically, these genes are upregulated in response to lifespan extending interventions, regardless of the type of intervention. And also we had ribosome protein genes here. So on the downside, we had mainly genes involved in immune response, such as complement and coagulation cascades and immunodeficiency. So the next idea was to see how consistent these changes are across different tissues. As I said, we had liver here, but our tissues just have less data sets available, but some of them are still available. So we wanted to validate these results using our tissues, and we could find enough data for two more tissues, for skeletal muscle and for white adipose tissue. So we ran the same analysis, and what we found is that at the level of individual genes, there were almost no overlaps. So here on Venn diagrams, you see almost all overlaps equal to 0. But at the level of functional enrichment, which is shown on panel E, we have a lot of similarities. So it means that basically different individual genes are perturbed in different tissues, but these perturbations lead to the same cellular changes at the level of pathways, at the level of groups of genes. And here we again see upregulation of oxidative phosphorylation, ribosome protein genes, metabolism of amino acids, etc. Well, not surprisingly, drug metabolism was activated only in liver. This is kind of expected. And on the downside, we again saw immune response related genes. So this was basically our first set of biomarkers. So these genes demonstrate if certain intervention leads to lifespan extension, but here we do not account for the effect of this intervention. I mean, here we basically every intervention has the same weight. You have growth hormone mutants, which extend lifespan by, let's say, 50%. And we have here in the same set, we have, for example, proton dim, which extends lifespan only by 7% and only in males. But here we are accounted equally. So to account for the size of the effect, we basically looked for genes which are associated not only with the intervention, but with the effect of each intervention. So basically, yes, here such genes are shown. So for example, let's look at panel C. Here we have a gene called HINT3, and on the x-axis, there is a change of maximum lifespan in logarithmic scales, logarithmic of ratio, and on the y-axis you have fold change in logarithmic scale as well. And here you can see that on average, the higher activation of the genes, the higher this gene activates in response to intervention, the more is the effect on the lifespan. So this is a positive association. For example, for the gene IRF2, which is interferon related gene, you have the opposite picture. So the lower activation you have, the higher is lifespan. So it's the opposite effect. And basically, this heat map on panel A shows all our findings. This is for the association with maximum lifespan. So you can see that some of the genes, they kind of increase their expressions. Our genes, on the opposite, decrease their expression. And here we don't show, but basically we also found one of the gene which showed one of the most significant negative associations with change of lifespan, was at the same time found to be one of the strongest biomarkers of mortality in humans. So basically, we found it after we already sent our final version of paper. So you can't find this info here, but this was also a pretty nice result. It's called Orza mucoid, I believe, something like that.
And what does it code for? What does it?
This is the name of a protein, Orza mucoid, and
right. Oh, right, yeah.
And it seems that the complete function is unknown, but it's related to acute phase inflammation. So it's basically an inflammatory gene. And this gene is specifically secreted in liver, so it's expressed only in liver. And then it goes to the bloodstream, and it was shown that it's one of 4 main biomarkers of mortality that can be found in bloodstream. So our 3 are mainly some physiological markers, such as the level of the number of leukocytes and something else. So this is the only protein that was linked to the mortality. Also interesting result we found here is that that the genes that are targeted by some interventions are not always associated with lifespan extension. So for example, it's known that DGAT1 deletion increases lifespan. DGAT1 is basically the gene of fat synthesis. And if you just delete this gene, it increases lifespan. So on panel B, if you look at the expression of this gene, you can see that basically it's downregulated only in response to DGAT1 deletion, not surprisingly. In all our cases, it's even slightly upregulated. So the target is not always associated with the effect. So targets are kind of master regulators, which seem to change the whole profile of gene expression, and then it kind of stimulates some changes associated with lifespan extension. So what we also did, we obtained the list of genes which manipulation links to lifespan extension. We obtained it from GeneH database, and we basically looked if these genes are associated with lifespan extension in our data, and basically we observed no correlation. So again, what we found here are not targets which you can manipulate to extend lifespan, but these are biomarkers. So this is systemic biomarkers. It's not targets. So it seems that these are 2 different concepts. Yeah, another interesting thing is that several biomarkers were found to be both common signatures and signatures associated with the effect on lifespan. So for example, here you can see NQO1, which is one of the targets of NRF2, acute stress regulator. And you can see that it's, first of all, upregulated in response to different interventions. And at the same time, it's positively associated with the effect on lifespan. So it's kind of very consistent signature. And another example is SLC15A4, which is the transporter in lysosomes, which is important for pH level. I mean, for the stability of pH level in lysosomes, and it also shows It's first of all commonly upregulated in response to different interventions, and it's also positively associated with the effect on that. So these two are a nice example. There are others, but not too much of them. So we could find something about 15 such genes, which are signatures of two types, both qualitative and quantitative signatures at the same time. Now panel C shows functional enrichment for these signatures associated with the effect on lifespan. So this is a similar analysis we had for qualitative signatures. We repeated it for quantitative ones. And basically some of the functions, again, turned out to be both qualitative and quantitative predictors of lifespan extension. For example, oxidative phosphorylation is again upregulated, and it's associated with all metrics of lifespan extension you can imagine. We had maximum lifespan, median lifespan, and hazard ratio here, and it shows significant positive associations with all three metrics. Also, you can find amino acid metabolism here again, and on the downside, again, immune response. So these functions seem to be pretty consistent both at qualitative and quantitative level. Well, panel D shows how similar are signatures associated with different metrics of lifespan, and basically here we just show that the overlaps are significant, and it means that basically regardless of the metric you use, you still obtain similar results. So just also kind of verification to be sure that metric doesn't matter. And finally, panel E is probably the most important panel in terms of practical usage, because here we applied biomarkers we obtained in our paper to predict new interventions with potential lifespan extending effect. So as I showed previously, our biomarkers are not targets which can lead to lifespan extension. So it's not that simple, like you just, delete certain gene, which we found, and voila, you have lifespan extension. It's not that simple. So you have to compare against the whole set of biomarkers. And here is what we did. We basically used similar to GCA approach, which we use for functional enrichment can be used here. So here on panel E, we have 6 different signatures. So one of them, the first of them is common signatures across interventions. 2 signatures are signatures associated with the effect on lifespan, with the effect on median lifespan and effect on maximum lifespan. And then we have 3 signatures associated with individual interventions, caloric restriction, rapamycin, and growth hormone deficiency. And on the y-axis, we have different interventions which were obtained from publicly available datasets and from CMAP predictions. CMAP is a well-known broad tool which basically shows how similar is your gene expression profile to chemical compounds, to the effect of chemical compounds on cell lines. And typically we have cancer cell lines there, so it's not really like biological model, but they aggregate, they basically have data on about 20 different cell lines, so you can look only at changes which are consistent across cell lines, so to be sure that they are not driven by cancer nature or something. So let's start from public data here. So basically we went to Geo database and we obtained certain data sets corresponding to interventions which have not been shown to extend lifespan, but have kind of hypothesized to have some effect on lifespan, and they are shown to have some effect on health span. So these are not just random interventions. And for example, we had interleukin 6 injection, and interleukin is related to inflammation. So we expected negative association, and that's what we saw. And the signatures were very consistent here. The next intervention was MET1A knockout, and MET1A is one of the genes involved in methionine metabolism. So if you delete this gene, the level of methionine goes up, and methionine reduction increases lifespan. So it's kind of opposite things, and this is also what we see here. So 5 out of 6 interventions demonstrate negative association with this intervention. On the positive side, we had hypoxia, chronic hypoxia, and KEAP1 knockout. KEAP1 is an inhibitor of NRF2. So if you knock out this gene, NRF2 goes up, and NRF2 is basically associated with drug metabolism, cytochrome P450s, etc. So it's kind of good thing. And here we also observe positive association. And then we had 3 different sirtuin activation models. So one of them, SRT2104, is a compound, and its effect on lifespan is not very high. And here we also observe that it's positively associated with some signatures, but not all of them. And the second model is overexpression of sirtuin gene, and most interestingly, it shows the effect only in males. And quite consistently, we observed the effect on signatures also only in males. So in females, we observed no effect, no significant effect on signatures, and no significant effect on lifespan and health span as well. Yes. So yeah, and another interesting thing we also had here is we downloaded data set which had gene expression of 2 different mouse strains of the same chronological age, but with strains differed in lifespan. So one of them was more short-lived, another was more long-lived. And you can also interpret basically genetic strain as intervention. I mean, basically 2 strains of mice, they differ in genetics, and basically genetics is also kind of intervention. And here our signatures were able to predict which strain lives longer. So it works in this context as well. Interestingly, in case of caloric restriction in rhesus monkey, this is also liver data, caloric restriction signature showed strong positive association. So it means that kind of caloric restriction effect is pretty similar on gene expression level across species, comparing mouse and monkey. But for some reason, we didn't observe any significant association with biomarkers of lifespan extension. So there can be, of course, a lot of hypotheses explaining that, starting from biological and I mean finishing with some technical explanations, size of sampling, etc. But I mean, this is what we observe. At least caloric restriction signature demonstrate that the effect of certain intervention is consistent across species. So at least we observe this effect. Regarding CMAP, what we did, we basically used this platform to predict drugs with the highest expected association with the signature corresponding to maximum lifespan. Because we were interested in drugs with the strongest effect, I mean, which extend lifespan by, significantly. So we used this signature, and then we chose 4 drugs from the top ranked list, which already have convenient designs for experiments in mice, and which can be orally supplemented to mice. And then we just gave them to mice for 1 month and looked at gene expression changes, um, basically in response to these chemical compounds. And what we observed is that CMA predictions pretty well predicted real biological changes. And especially in case of first two drugs, we observed consistent positive association with almost all signatures. In case of our two drugs, the association was significant for signatures associated, common signatures across interventions, but not with signatures associated with the size of the effect. So it kind of predicts that the effect, there may be an effect, but it's not very high. And interestingly, one of them is also mTOR inhibitor. But for the first two, basically the prediction is very nice, and now we run longevity studies to observe if in direct experiment we really may extend mouse lifespan. So this is the idea. So in general, basically the main aim of our work was to obtain the set of biomarkers which can then be used to simplify and accelerate the search of new interventions. That was the idea. So I guess that's mainly it. Do you have questions?
Yeah. Are we stopped sharing the paper now, or are we still we have it up?
As you want.
Get our faces back up for the world to see. Yeah. That was a ton of work, and definitely would be very useful for people who are searching for new interventions and techniques. One question that obviously pops out is, its applicability across different species. Obviously, there's correlation there when, you utilize data from rhesus monkeys, but a lot of the data is from mice, and some of the interventions obviously seem to have a, like CR, for example, more and more negligible effect on longer lived species. Although, that might still correlate with an extension of health span. And like you said, it may have something to do with the size of the data set as well, because you might eke out, a little bit more lifespan, but that, you might have to have a lot more monkeys in the study to kind of see that effect. But you might have a more pronounced effect that's visible on loss of morbidity, which is indeed that was seen. So I guess what everybody's interested in is its applicability to humans, because that's we'd like to see that. And I'm just wondering if this type of analysis that you're doing here, is it applicable? Is some way you can apply this technique to different sets of humans, for example, centenarians and supercentenarians, and people where it's predicted that longevity, let's say, runs in certain populations or families? Can you apply this technique, and say, okay, well, this is a signature. We would predict that, this group of people, with this type of either this type of lifestyle or this type of genetic background would have this signature that is indicative of increased longevity versus another human population. Have you applied this technique?
Not yet. We didn't work with humans, but theoretically you can do it. The problem with humans is that you don't have really good models, because even if you take centenarians, well, you will probably observe a lot of signatures associated with the age, not
Right.
with the last effect.
Right. Obviously, the best thing to do would be a longitudinal study, which would take a long time.
Yeah.
And the second best thing would be basically, looking at populations or where there seems to be, a genetic link between certain families or certain, and get enough people, where you can make, I guess, have enough population where the data could make some statistical sense. I guess that's the second best thing that
Oh
you can
yeah,
do.
yeah. I think it may be done. Um, the data is the problem, but so theoretically, yes.
Okay,
We don't have such data, so that's why, I mean, in our case, it's almost not possible.
right,
Uh,
and yeah,
what probably we can do is if we get data on different lifestyles, as you said. For example, I don't know, the effect of low-fat diet, high-fat diet, or exercises, something like that. We can look if these changes are at least consistent with the changes we observe in mice.
well, the simplest intervention right here would be hypoxia, right? People living at high altitude, low altitude, you should see a similar type of.
Uh, well, maybe. I'm not
Right.
sure because in mice, when we apply chronic hypoxia, there is not yet any physiological changes. So in humans, maybe the people who live, I mean, high in mountains, they already adapted to these conditions, so they may not have these signatures.
Right. Well, I mean, there's ways around it, right? I mean,
Yeah,
people move and migrate. So obviously, if you have a population that's been there for 2,000 years living on top of a mountain, then there's going to be genetic
yeah.
selection. But people have just migrated from high altitude to low altitude. There should be ample
In general,
population.
I guess you can do it, and it's probably will be similar to what Steve Horowitz did at the level of methylation clock when
Mhm,
he looked how it changed in response to different lifestyle factors.
mhm.
So probably the same can be done in case of gene expression. The problem is that our analysis is done on livers, so
Right.
you you must have that person for that. You cannot take liver sample from one person.
Well, you could probably painful. Liver regenerates pretty quickly, but mm, you you could not easily. No, no, yeah,
Yeah,
I agree with you. It's much easier than a brain biopsy, but definitely not as easy as
with
a
the
skin
brain, it's
biopsy.
interesting that we don't show it in paper, but we also had brain samples in case of our own RNA-seq,
Hmm.
and we didn't include it in paper because we observed no changes at all. So it seems that in brain, almost no changes occur. And but the only interventions that induced changes in brains were mutants, which is again uh, kind of
Really?
probably not surprising because seems that just chemicals do not pass through the barrier, so you don't observe any effect. But in case of mutations, they occur everywhere, so you
What?
still might
Well, what about dietary interventions?
a
No.
very small, very limited effect, almost no effect.
And is this potentially due to? I mean, obviously the neurons, different types of cells, but I mean, the other major difference is, I mean, fat cells and adipocytes and hepatocytes. They replicate. Most neurons don't. You had muscles here, which I guess the muscles that you're using here, they're not going to be. Most of them are not going to be dividing, right? So
Mhm.
we're looking at. So I mean, is there a difference between expression profiles that you look at between primarily dividing and non-dividing cells?
Yes, there is, and we also had in our lab we had a paper on differences between, I mean, associations between gene expression and cell turnover, which is exactly
Mm-hmm.
what we're talking about.
Mm-hmm.
But in this paper, it was done not by me, but basically I had the results, and they didn't correlate with my biomarkers at all. So they were totally different. So what we see here cannot be explained just by the change of cell turnover.
Right. And kind of, I add another question about, we don't have to go back to the figures, but you looked at a lot of interventions and two interventions, and I'm thinking most of these were, from mouse cells. You had two mouse models, I believe, of Huntington's and Parkinson's, right? And is that correct? Uh,
No, no.
those are human?
We didn't have
Uh,
neurogenetic models.
what?
These were Huntington's and Parkinson's were in GCA there
Oh,
are
okay.
gene sets
Okay.
associated with diseases, but this is just annotation from
Okay.
yeah.
So the upregulation of genes there that correspond with lifespan. I mean, is that because of are those gene sets sort of like forming a protective function? Is that the hypothesis?
Yeah, so the upregulation of these functions doesn't mean that, I mean,
Yeah.
these changes you can also observe in these disease models. It
Right.
means just that the genes which are associated with which are basically elements of the pathway which is perturbed by these disease models are upregulated. But this can be both defensive genes and really harmful genes. So it doesn't tell you about the health span,
Right. Right.
but it tells you that basically what we observed is that three aging-related disease models were associated with the effect of longevity interventions. So it shows the link between basically the aging diseases and lifespan extension, which is also kind of expected result.
Hmm. Any questions from? Do we have any questions on the chat? Anybody?
I think we've blown their mind, which is a bit worrying by the sound of it. But from what you were saying about the brain,
Uh-huh.
it's a little bit worrying if we can't renew the brain. We should try and avoid blowing the minds of the audience. But yeah, there's a lot to think about there, isn't there? I noticed that Cert 6 was mentioned.
Mhm.
Cert 6 always comes up. Everyone always talks about Cert 6. David Sinclair and Vera Gorbanova. It's it's activated or dependent on NAD plus anyway in order for it to operate. There was, you recall the paper that we did, didn't you, Oliver?
Hmm.
Vera's paper where we talked about the association of sirtuins
Hmm.
with longevity. So it's quite interesting. Yeah, there's a lot to think about there, but
So
yeah.
where can we find the Gentervention tool? You have a link there, correct?
It is online in paper. Yeah, you have a link in paper. It's very easy to access.
Ah, okay.
Yeah.
Is there we can't is there any way we can put up the link here? No, we can't do that easily, can we? There.
I can put it in the chat.
Okay. All right. And everybody has access to the chat, correct?
Yeah, I'm just going to Google it. There we are. So if people are interested in actually using the Gentervention platform, I'm going to use that word. I've put it in the chat there, and that's free to use, I believe.
Yeah,
Mhm.
sure.
That's a great little resource. I suppose the next step must be larger, running this approach with more and more longevity or alleged longevity interventions to see what else might come up.
So I guess one comment I have to make, sort of a big picture comment based on the data that you showed, and you had kind of grouping of three different major interventions and their and the functional outcomes of the three, being genetic interventions, dietary and pharmacological, and then basically depending on the intervention, the effect on longevity or the effect on these functional outcomes were different, right? So you had a more robust effect with some of the genetic interventions, less of an effect with dietary, and least of all with pharmacological.
Mhm.
And I'm assuming the pharmacological were individual pharmacological interventions, like a single drug,
Yeah.
like rapamycin.
Yeah.
And it's interesting because I'm just, based on like all the other papers that I've read. I mean, correct me if I'm wrong. I'm just trying to,
Mhm.
hypothesize here how this would fit into the kind of grand scheme or framework of how this works. If you look at some of these genetic interventions like growth hormone receptor knockout mice, et cetera, you've got pretty radical genetic interventions that disrupt a master regulator. So you have a lot of downstream effects.
Mhm.
Dietary is very multimodal, meaning that you when you change your diet, you're taking in lots of different compounds. You're altering, your caloric status, and you're targeting a lot of pathways also at once. And then you get to pharmacological, which are targeting individual pathways. So this almost suggests to me that if you want to have as a robust effect as some of these major genetic interventions which target a master regulator, or even a dietary effect, with a pharmacological effect, you most likely won't have a silver bullet drug, right? You might have to target several orthologous pathways using several different pharmacological interventions all at once. That's the suggestion I'm getting from this data. Is this, you think, an accurate assessment?
I think it's pretty reasonable, yeah. What we observe in every our study is that aging is a systemic process, and if you want to really slow it down, you should slow down all different pathways associated with that.
Right. So there's an interesting. This was just came out, and I haven't read the paper, but there was a clinical trial that was actually done in the UK in conjunction with a lot of villages in Iran, which looked at a polypill, right? And this had nothing to do with longevity per se, but had to do with cardiovascular mortality. And I think that the pill had like 4 known interventions combined at once. It was, I think, salicylic acid or aspirin. It had I think it had a statin, and it had 2 other blood pressure lowering drugs. So it was like 4 things all at once. And they looked at the mortality rate of thousands of people, and then they compared placebo, actually the placebo was they gave dietary and lifestyle advice versus dietary lifestyle advice plus this poly pill. And the difference was, at the end of this trial, Group A had 300 some odd people die, and Group B with the poly pill had 200 some odd people die. So we had like a 35% decrease. So basically the numbers would be that, you would, at the end, and I think the trial was like 5 years. So at the end of that period of time, you would, it was significant. It was pretty significant. And so I'm, going back to all of this aggregate data here, that's something that people talked about for a long time. I know David Sinclair's talked about a polypill idea for aging, and certainly it, all of this data suggests that a sort of multimodal if you're going to do a pharmacological intervention, to get as a robust response, you would probably have to target multiple pathways at once. And I think there was a C. elegans paper we did a way back, which suggested that there was a synergistic or additive effect of targeting at least 2 or more pathways for C. elegans lifespan. And I forgot what the interventions were. I think it was, I can't remember.
It was shown in mice by Richard Miller, I guess 2 years ago or 1 year ago even. So they had another paper related to ITP program where they test interventions on big samplings of mice, something like 150 mice, in 3 independent laboratories across US. And they showed that rapamycin plus metformin together increased the lifespan stronger than each of them individually. I mean, even after addition, so very synergistic effect. So in mice, it was also shown to be the case.
Yeah. So I think that's some pretty to me that's some pretty important information, to add to kind of the arsenal of looking for drugs that would significantly extend human lifespan. The question is how much lifespan versus health span, extension will we get with these interventions? Because the best that we'll probably get is a mimicking of dietary interventions, which is still significant. I think how much of this is going to trend to, I guess the bottom line is, we're probably going to be looking for a combination of drugs that makes the majority of people live to or equal the health span of the people that are the lucky few that end up being super centenarians or centenarians, right? So that would probably be what we'd be looking for. And I don't know how doable that would be with a pharmacological
This
mix.
is pessimistic
That's the hope.
scenario.
Hmm?
This is pessimistic scenario. I believe that we can
That's,
go and top it.
yeah. So that's not a bad pessimistic scenario, right? I mean, to live to 100, 120 without signs of dementia or any, and then die peacefully in your sleep. That's a pessimistic scenario, and that's not a bad pessimistic scenario. And if can pharmacological interventions in combination achieve beyond that, I'm personally doubtful, but small molecules, but it certainly at the very least could buy us a lot of more additional precious healthy time.
Yeah, and that really ties in with what Vadim Vladishev has recently said at Berlin about the three. You remember this, Oliver, about the three types of interventions. You've got your geroprotectors, you've got your repair approaches like SENS, and you've also got your cellular or partial cellular reprogramming like OSKM, etc., OSKLN, and there's a few different ones that people are experimenting with. So it really ties in with that. I think you're right about small molecules. I think we could probably get some mileage out of geroprotectors, and they certainly have their place. I totally agree with what Vadim has said about all three of those interventions having their place. But I suspect you probably are right. I don't want to be pessimistic, but I think
I mean, like I said, I mean, the what this data to me suggests is even the pessimistic outlook is pretty damn good, right? So if what this suggests to me is that if there is no magic pill, there is probably a magic poly pill that will significantly extend the health span of individuals. So I'm pessimistic about a magical pill, but I'm really optimistic about I'll just use the word magical without magical combination, right? That basically targets orthologous pathways or slightly orthologous or some path, there's going to be crosstalk, but that could mimic a kind of a robust genetic intervention or a robust dietary intervention, right? So it's certainly the data in this data here and the data in C. elegans and additional data out there suggest that that would work. Probably won't make you live to 1,000, but the data suggests it will make you live healthier longer, and that's a step in the right direction.
Yeah, sign me up. I'll take 120.
And at the same time lower the lower your risk of all diseases of aging at the same time, right? So that's pretty huge.
Yeah, I mean, anything that the buzzword these days is health span, of course, because there's absolutely no point extending lifespan if you're gonna be decrepit, and that's definitely not the aim of the field anyway. People often say, oh no, it's going to be another 20 years in a wheelchair or a nursing home. I don't know any researcher working in this field who says, you know what I'm going to do today? I'm going to find out a way to extend just lifespan, not health, just lifespan. And yet people always seem to have this misconception about what the field is trying to do. At the end of the day, it's about healthy, longer life, right?
So another question I have for you, Alexander, is you, when you applied this analysis, you came up with these identified 4 compounds with significant effects, and then you applied them to the mice. 2 mTOR inhibitors, 2 other compounds. Now these were then guess, tested, and some of these were actually known for a long time, like ascorbyl palmitate. The reference is there to 1974, which is my date of birth. Uh, can this analysis help figure out potential drug combinations, right? Because if you're looking at a profile that's going to extend lifespan and you're coming up with these compounds, you probably want to test which combination would work, not just individual drugs.
Exactly, but that's basically the idea of our current project.
Oh, so that's the current project right now.
Yeah,
excellent.
Yeah, so the idea is to, first of all, to predict not only the statistical effect, but the real quantitative effect. And this will allow to predict which combinations can give the most synergistic effect.
Okay, so I hope I didn't let the cat out of the bag. I hope I hope this is not top secret information. So if you don't want to talk more about it, but I'm sure you're going to have some awesome data, based on other papers that have suggested that would indeed be the effect. It would be very interesting to see what certain combinations have on synergistic effects on lifespan of mice and other and other organisms besides C. elegans and other short-lived organisms. So I will definitely be looking forward to that data.
Yeah, and another thought is Senolytics as well, because it's increasingly clear that Senolytics is probably going to need a cocktail approach. Peter de Keyser and others have already talked about senescent cells using different pro-survival pathways, like the BCL2 and various other pathways. And the problem is no one small molecule targets them all. So that's a problem. We're actually experimenting with combinations at MMTP of different known or alleged senolytics. So it'd be interesting to see that platform developed.
Did Alexander just disappear?
Well, I hope that wasn't a senolytic effect, but but yeah, but that could be quite useful anyway, couldn't it, Oliver?
Yeah. Oh, I had just a great question for Alexander here before he blipped off the radar. But anyway, before it escapes my mind here, and Alexander, if you're listening out into the ether out there, or if you come back, but anyway, we're recording this for posterity. Regarding senolytics and regarding the hallmarks of aging, the hallmarks of aging, where we look at telomeres and telomere shortening and senescent cells and all of the proteostates, all of the other things that form these hallmarks of aging. It's real. I mean, these hallmarks are real, but the categories are also, there's maybe some arbitrariness, and people are actually using this review paper and the hallmarks as ways to target individual boxes, right? Saying, okay, my intervention targets this. Oh, there you are, Alexander. So I was just talking about hallmarks of aging, and I had a question for you, and this ties into the paper or the work you're doing now with synergistic effects. People looking at compounds, are approaching hallmarks of aging as if they are real, distinct effects. So 9 hallmarks or 7 hallmarks, or there is some arbitrariness to it. With your approach here, I'm thinking, if you want to get, so I guess what I'm getting at is, if we want to come up with a combination that is, let's say, the most robust combination pharmaceutical intervention to theoretically maximize lifespan and or health span, right? Your intervention can suggest how many orthologous pathways or which ones to hit, right? So you should be able to predict, theoretically, or you should be able to look at your data and say, okay, that's it. With these four pathways, or these 5 pathways, or these 3 interventions, or 4 interventions, we're hitting a theoretical maximal plateau effect of we can't squeeze out any more effect. with these drugs, you know, versus, you know, and I don't know what that number is, or I don't know what all those pathways are, right? Versus, let's say, taking the hallmarks of of aging, which is a useful guide, but somewhat crude, because it's based on looking at papers and aggregating a lot of data. You know, let's say there's 9 hallmarks of aging. You know, that doesn't mean that coming up with 9 drugs that target each of these hallmarks is your theoretical maximal effect for a pharmacological intervention. Your approach here would suggest that to me. Is that a fair assessment? If you're looking at a synergistic effect, I mean, there will be a certain, in my opinion, saturating effect, right, of optimizing pathways beyond which you can't optimize pharmacologically anymore.
I think it should be, but we do not observe it with this data at least. So we were looking exactly at what you're talking about. We were looking at some discrete number of pathways, which kind of group separately, and very limited number of them. But we in fact couldn't find such clusters of genes. So
right, like they might, right, like I don't like, for example, there might be an upstream regulator of 2 of the hallmarks of aging, right, that one pharmacological intervention might target. So you might end up targeting hallmarks of aging with less compounds, for example, and the model might predict that based on how much crosstalk there is between pathways.
Oh yeah. Even with upstream regulators, we don't see very limited number of them. So we did such analysis for caloric restriction growth hormone deficiency at rapamycin, and we identified something like I don't know, 50, 60, or even hundreds. I don't remember exact numbers. But so again, it can be explained just by technical explanations. I mean, a lot of upstream regulators have common targets, shared targets, so they kind of overlap with each other. You cannot really separate.
Right.
But well, we believe that the easier approach would be just to combine individual effects than to try to find some upstream regulators. So what we want to do is, for example, we have, let's say, 100 genes, and one of the intervention is shown to regulate 50 of them, but another is shown to regulate, let's say, 70 of them, and 20 overlap. So if we combine them together, then the whole set of 100 genes will be covered. So this is the idea.
Right.
It doesn't take into account the upstream regulators, but it should be easier to implement. So we want to start with this approach. Yeah, talking about the limit of lifespan, we have another project which is more theoretical. It's basically it's about theoretical model of Gompertz curve, and it's basically it's shown that the Gompertz curve has 2 parameters. One of them is kind of initial rate, and another is rate of mortality, but these 2 seem to be correlated with each other, so there is a dependence between them. So in reality, you have basically 1 parameter, and with this 1 parameter model, there is just theoretical maximum of lifespan you can achieve, because at some point then you decrease mortality rate, you increase initial rate, and at some point there is an optimum level. So they have such work, and with that we were able to predict maximum lifespans of different species in response to interventions, and these predictions, though pretty well fitted to the real data, I mean, to the maximum possible achieved in experiments lifespans. And we also did the same analysis for humans, and based on that we predicted the maximum limit of lifespan which can be increased by interventions to be around 150 years. So it's 30 years more than current records, but it's still limited. So it's not, I don't know, 1,000 years.
I'll take that. I'll take 150 as a starter.
Okay.
Yeah,
That's not bad. That's not bad at all.
as Aubrey de Grey says, so if we have time till 150, we will be able to already find new solutions.
I mean, that's incredible, really. I mean, taking the global average lifespan, life expectancy, I think it's around about 75 without checking the data.
Yeah.
So 150 is incredible. It's nearly double.
Mm-hmm.
That's
But
incredible.
this is maximum. It doesn't mean everybody will live 100.
Yeah. Still, that's
We can't all be Jean Calment, can we?
Still, I just want to point out to the audience, this is the pessimistic account.
Yeah.
So that's so we got to. So that means that as far as the roadmap with current technologies and current pharmacological interventions and the road that we are going on right now without any kind of radical breakthrough, there's a lot that we can achieve right now with the kind of the tools in our in our toolbox right now. Who knows how much further we can achieve when if we break past the 150-year barrier with new technologies that are just beginning to be utilized or as yet have not come across the horizon. So there's a lot of work to be done right now that could positively affect our lifespan.
Yeah, there's a lot of mileage, it seems, that we can get from small molecules, which is very interesting. And I'll take 150. But yeah, I am very interested in things like partial cellular reprogramming. The sky is perhaps the limit. So, but yeah, I'll take 150. I'm okay with that.
Anyway, I'd like to end the meeting at this note because I have to take off. But here's to, I guess, a healthy 150 years in the short term for everybody and beyond. And I'd like to thank Alexander, our newly minted Dr. Tyshkovskiy, for presenting this wonderful paper, taking us through the data, and also for giving us a glimpse as to the experiments that are taking place right now. That he's let us take a peek on, which we'll be eagerly looking forward to, and no doubt doing another journal club in the near future once that data is made publicly available. So thank you very much.
Thank you very much. Thank you. See ya.
Yeah, that's great. And speaking of thank yous, I'd like to obviously do the usual and thank the Lifespan Heroes, who are our monthly patrons who help make this possible and many of the other things that we do. If you're interested in supporting us, do check that out. It's lifespan.io/heroes. And I guess we'll see you a similar sort of time next month, Oliver, I suppose. We tend to make it a regular sort of thing, don't we? And who knows what we'll be looking at next time. So,
all right.
Suggestions welcome.
Yes, stay tuned.
So you know where we are. All right. Thanks very much.
All right. Thanks, everybody.
Thank you for the invitation. See ya.